Hello everyone, thank you for joining me today. My name is Noa Moriel and I'm a PhD student working at IBM Research. Today I'll present to you our work on evaluating how well transcriptomic foundation models recognize real gene expression patterns at the sample, cluster, and dataset levels through null sample testing. Okay, so let's begin with the core idea. Transcriptomic foundation models like SAGPT are pre-trained on diverse single-cell RNA sequencing datasets and have shown strong performance across downstream tasks. Training loss is a standard measure of model performance. However, it is challenging to interpret since data statistics vary. It's unclear what is a good value. Is 1, 10, 100 a good value? And this is especially non-trivial for out-of-distribution datasets. Here we propose comparing the model's pre-training loss on real data versus synthetic alternatives. So our hypothesis is that a model pre-trained on gene expression data should have lower loss on real cell states compared to fabricated ones, like shuffled or interpolated samples. And this is analogous to language models where we would expect the shuffled sentence to yield higher pre-trained loss than the original sentence. So let's see whether the pre-training loss reflects the model's familiarity with biologically plausible expression patterns. Okay, so to evaluate this, we use SAGPT pre-trained on human single-cell data. We compare the quality of the model's predictions on 3 types. So first, the original unmodified expression profiles. Then we have the shuffle data, where we shuffle the gene identities. And last is the interpolated data, where we look at linear combinations of profiles according to the cell type origins. And in this schematic, we show our test is the loss on the real data consistently lower than that of the shuffled or interpolated data. So in the XY plane is the gene expression space, and each point here is a cell or gene expression profile, and the black points are the original data. And in the z-axis, we depict how we imagine the pre-training loss over the samples. So being higher where the samples are not real ones. So either in purple the shuffle data or in teal the interpolated data. Okay, so first we look at the shuffled case. So at the top here we see an example of how we shuffle. So we just shuffle the gene identities, as I said. And at the bottom here we have the results. So on the left is the evaluation of the pre-training loss, and on the right that's an evaluation of the cell embeddings. So let's start with the pre-training loss. In both of these cases we evaluate on datasets from SC-eval, and we just take a single batch as a representative case for each dataset. So on the left here we compute the difference, the gap between the shuffled and the original data pre-training loss. And what we see here is that in many cases the model assigns significantly higher loss to shuffle data, suggesting that it can distinguish real data from fake data, but it's not always the case. So in some of the samples here, the shuffle data gets as low or even lower error than the real data. So then we were wondering whether the cell embeddings still reflect the cell types after the shuffling, or are they also impacted by the shuffled values. So to evaluate embeddings, we train a logistic regression classifier on the cell embeddings to predict the cell types and use an F1 score to evaluate these embeddings. The dark red mark is a stratified baseline that just randomly samples the labels. So what we can see is that the shuffled embeddings lose their predictive power. They no longer separate cell types effectively. So this shows that while models are often evaluated by how well embeddings align with biological labels, they also show promise in discriminating real versus fabricated inputs. So that offers a complementary view of what the model learns, but it's not perfect in discriminating between real and fabricated inputs, and that can be improved maybe in other models. Okay, so this was for shuffle data, but shuffle data can be far from biologically realistic samples. So here we also explore the model's behavior within the gene expression manifold. So we generate interpolated samples between cell types. These interpolations probe the boundaries between gene expression states. And in these PCA plots, we can see the original data in black, and the color points are the interpolated samples. So, for example, in the heart atlas, we see a smooth gradient from orange to blue, representing interpolated samples between the fibroblast and the cardiomyocytes cell types. Okay, so to quantify the model, how the model responds to boundaries, these transitions, we compute a log ratio. Specifically, we look at the pre-training loss of the interpolated samples versus the expected loss based on its parent cell types. So when the log ratio is higher than 0, then there is that signals a barrier or a learned distinction between the expression states. Okay, so what we see here in these 3 cases is that we observe such boundaries across many of the cell type pairs, but few reach statistical significance. And this raises interesting questions like, why are some transitions well learned while others are not? Are these differences driven by biology, by data imbalance, or by the training regime? And whether improving the learning of these boundaries could enhance how embeddings represent cell states. Okay, so to summarize, we see that the pre-training loss can serve as an unsupervised signal of how familiar a model is with real data at the sample, at the cluster, and at the dataset level. It complements embedding-based analysis and offers insight into model behavior under perturbation. Moving forward, we aim to link these findings to downstream performance, explore training strategies that encourage a structured loss landscape, and use pre-training loss as a confidence or interpretability signal, for example, to understand how well can we trust attention weights for gene interaction inference. All right, so with this, I would like to end and thank my collaborators at IBM, Michael, Ishai, and Michal, and thank IBM for supporting this research. To learn more about IBM's presence here at the conference, feel free to scan this QR code. And please feel free to reach out with any questions or feedback. I would love to connect. Thank you for your attention.